Hello and welcome to Nikolai's genetics lessons, and today's problem is a circular peptide is placed in extended condition. If total 50 amino acids are there in peptide, what will be the radius of peptide? And here's the answers to choose from. And actually this is very simple problem. Actually this is not as much genetic problem as a geometrical problem, and probably we study this type of problems in the 5th grade. So I was thinking, what is the purpose of this problem to ask students, and probably this is to teach you the scale that we usually work with when we talk about where the bases of the DNA or RNA or single amino acids and peptide that they make. So what do we call peptide? And we call peptide when we have two amino acids, for example, like in this example, that would be joined. And now we have a peptide bond here, and now we call this molecule dipeptide. That means that it consists of two amino acids. And if we add another amino acid here, we are going to have tripeptide and so on. So we will call small proteins peptides that have about 50 such amino acid residues. So what is the average length of one such amino acid? And this is 3.5 angstroms. So how small 1 angstrom? So imagine nano. Nano means 1 billionth of a meter. So 1 divided by a number with 9 zeros. So 1 billionth. 1, 2, 3. 1, 2, 3. 1, 2, 3. So this number represents 1 nanometer. And 1 angstrom is a number that is 10 times smaller than nanometer. So we have to add another 0. So 1, 2, 3, 1, 2, 3, 1, 2, 3. So 1 divided by 10 billion. So this is going to be angstrom. And this is going to be a nanometer. So if we know that one amino acid about 3.5 angstroms, on nanoscale this is going to be 0.35 nanometers. Once again, it is very easy now to make a mistake here. Let's for comparison use just a meter. So imagine that this is a meter, and here we have say about 50 centimeters, and somewhere about here we would have 10 centimeters. So 0.5 meters and 10 centimeters. So here we calculate in centimeters, and this number those bigger than this one, but we calculate in meters. So meter is bigger than centimeters, so as you see 0.5 meters would be bigger than 10 centimeters. So do not mess up here these numbers. 3.5 angstrom, we also put a circle on top of A, would equal to 0.35 nanometers. Nanometers are 10 times greater or bigger than angstrom. So now we can solve our problem, and probably one more thing that you need to know in order to solve this problem is P number, which is equal to 3.14. and we can find diameter of the circle if we divide perimeter by the p number. So let's return to our problem. According to our problem, we know that amino acid chain or polypeptide consists of 50 amino acids. So we have to multiply 3.5 angstroms by 50. And we are going to get 175 angstroms. And now, as you see, this is going to be perimeter. So this is going to be perimeter. And in order to find diameter, we have to divide this number by p. So diameter would equal to 175 divided by 3.14. And the answer would be 55.73. Number represents a diameter, and we need a radius. And radius is a half of the diameter. So once again, here is a circle, here is a diameter, and here would be radius. And radius, as you see, is 1/2 of the diameter. So we have to divide this number by 2. So divide by 2. And the answer would be 27.86. Now we can choose an answer. And as you see, we don't have exact answer, but answer B is very close to the number we have found. Probably the person who prepared this test made a mistake, rounding mistake. So actually, if we need 3 significant figures, we are going to get 28.0. So this is going to be our answer, 28.0, if we need to round it. So as you see, the correct answer would be answer B. Those probably is some mistake here, or maybe this number was received using slightly different numbers, for example, for the length of the one nucleotide. Because this number is also rounded number. When I am preparing even such easy problems like this one, I am still trying to find some new and interesting information, and probably this picture would help you understand the significance of the circular peptides. This peptides is almost something that you cannot find in the nature, because any peptide would have N-terminus and C-terminus. So usually these two terminus wouldn't make a perfect circle and wouldn't make covalent bonding here, because in most cases these two ends would be separated by the certain distance between them that prevents them bonding and making a circular peptide. But nowadays we have found that such peptides would be much more stable if we would close the ends. And this is very important, for example, for the stability of the some medicine, or it's also very interesting application to use in designing some proteins and enzymes that have to be used in the harsh environment. Especially when we design such enzymes for higher temperature, or if we need to use such enzymes in some solutions with high or very low pH. And here's some examples of such circular proteins or peptides. And as you see, here's a circle, here is a circle, and here is another circle, and here's more examples. This time this is graphical representation. It's not necessary that this is exactly the shape of the polypeptide, but this is just a graphical circular presentation of the sequence of these peptides, and as you see, this is circular peptides. Of course, such peptides would be much more stable than those peptides that would have free N and C terminus. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.